This is Chapter 3, Module 5, Transcription, Translation, and Protein Synthesis. The learning objective of this module is to describe transcription, translation, and protein synthesis. Each cell needs proteins in order for it to survive. These proteins are used to replace old or damaged structural cell parts, to protect the cell, to carry things, to respond to changes, and to catalyze reactions. Basically everything the cell needs to survive on its own. New proteins must be produced or synthesized on a continuous basis. Every cell that contains a nucleus will be able to make its own proteins. Within the nucleus are the instructions on how to make new proteins. These instructions are invaluable to the cell. These are the only instructions the cell has for its survival, and so it's very important that they are kept protected within the confines of the nuclear membrane. These original instructions must never leave the nucleus where they could be possibly damaged or destroyed. The proteins, however, are produced outside of the nucleus in the cytoplasm of the cell. So, copies of these instructions will need to be made. These copies can move to the cytoplasm while the originals stay protected within the nucleus. These instructions are called DNA, and the copies that are made are called messenger RNA. The process of making messenger RNA is called transcription. In other words, the instructions were transcribed or copied so that the original instructions could remain in the nucleus, protected by the nuclear membrane, while the copy leaves the nucleus through the nuclear pores to enter the cytoplasm. In the cytoplasm, a new protein molecule can be produced. The messenger RNA binds to a ribosome, and the process of translation occurs where the messenger RNA directs amino acids within the cytoplasm to combine together to form a protein. In other words, the language of the messenger RNA will be translated into a sequence of amino acids. This module will discuss both processes necessary for the protein to be produced, transcription and translation. To understand these processes better, we should take a closer look at DNA. Each DNA molecule contains segments called genes. A single gene contains the instructions to make one protein. Within each gene is a specific sequence of nitrogen bases. Remember that adenine, thymine, cytosine, and guanine are the bases for DNA. Each gene contains thousands of these bases that are arranged in a different sequence in each gene. The sequence of these bases is called the genetic code, and it determines what protein the gene will produce. On each gene segment, the bases are grouped in threes called triplets. One triplet provides the code to produce one amino acid. Since proteins are chains of amino acids, each triplet will code for one amino acid in that protein. For example, if a gene segment has 1,200 nitrogen bases, that means there are 400 triplets and 400 amino acids would be produced. The resulting polypeptide, or protein, would be 400 amino acids long. Transcription is the process of taking the original genetic code from a gene and copying it into a messenger RNA molecule. Transcription has 3 steps. 1. Gene activation. 2. Duplicating the DNA to messenger RNA. And 3. Processing the new messenger RNA molecule. We will begin with gene activation. Each gene in the nucleus has to be activated before it can start transcription. When the cell needs a specific protein, that specific gene will become activated. Gene activation is partly related to the uncoiling of that portion of the DNA molecule where the gene is. It is estimated that the human body has 20,000 to 25,000 coding genes. Each gene must undergo activation before it can provide the code for protein synthesis. The first thing that happens in gene activation is that a segment of the DNA begins to unwind and reveal the specific gene needed. Proteins called histones are released from the double helix formation, and the winding staircase structure begins to straighten out. The sequence of the nitrogen bases can now be easily seen. Each gene segment contains hundreds to thousands of triplets that contain 3 nitrogen bases each. One of the triplets on the gene is designated the promoter region, and this is where transcription will begin. At the other end of the gene is a triplet called the stop region, and this is where transcription will end. Once unwound, the base pairs in the gene are exposed. The hydrogen bond holding the complementary base pairs together are easily broken, separating the 2 strands of nucleic acids. These 2 strands of nucleic acids will be held apart by other proteins. One of these strands is designated as the coding strand, and the other strand is designated as the template strand. An enzyme called RNA polymerase will bind to the template strand and assist in making messenger RNA. The coding strand reveals what the new messenger RNA strand will look like, except for where the coding strand contains a thymine, the messenger RNA strand will contain a uracil. In other words, messenger RNA is a duplicate of the coding strand, except that uracil replaces thymine. In summary, gene activation involves the unwinding of the DNA molecule and the binding of RNA polymerase. The next step is transcribing the template strand to messenger RNA. RNA polymerase has already been bound to the promoter region of the template strand. This promoter region is a sequence of 3 nitrogen bases called a triplet. The RNA polymerase moves to the next triplet on the template strand, and transcribing begins. RNA polymerase attracts nitrogen bases from the nucleoplasm to the template strand where the new nitrogen bases will bind together through dehydration synthesis. These base pairs are packaged into nucleotides with a phosphate group and a ribose sugar attached. RNA polymerase will pull in cytosine to the template strand where guanine is located. For cytosine on the template strand, RNA polymerase will pull in guanine. For thymine, RNA polymerase will pull in adenine. But for adenine on the template strand, RNA polymerase will pull in uracil instead of thymine. Remember that the new molecule being produced is an RNA molecule and will not contain thymine, but instead will contain uracil in its place. RNA polymerase is just large enough to cover one triplet on the template strand at a time, so when it slides down the RNA, it will move from one triplet to the next. Here it will attract the appropriate base pairs from the nucleoplasm to the corresponding complementary bases on the template strand. These nitrogen bases from the nucleoplasm are attracted to the complementary base pairs on the template strand, but they won't bind to the template strand. Instead, the newly attracted nucleotides bind to each other in the process of dehydration synthesis. The 3 newly bound nitrogen bases are called a codon, and they are complementary to the triplet on the template strand. After the nucleotides of each new codon are bound, RNA polymerase moves to the next triplet and another codon will be formed and bound to the growing messenger RNA strand. The elongation of messenger RNA will continue until RNA polymerase reaches a specific triplet on the template strand that codes for a specific codon. This specific codon is called a stop codon, and when it is produced, it will trigger RNA polymerase to break away from the template strand. At this point, the new messenger RNA molecule is floating free from both the RNA polymerase and the DNA. When this happens, the DNA molecule will zip back up by forming hydrogen bonds between the complementary base pairs of the template and coding strands. Before exiting the nucleus, the messenger RNA has to complete one more step. It needs to be edited in a process called RNA processing. The new messenger RNA molecule may contain unnecessary codes within it that can be removed. These unnecessary codes aren't needed to produce the new protein and would cause mistakes in making the new protein. They are called introns, and they are removed from the messenger RNA strand. The remaining codes are good codes that will all be used to produce the new protein. These remaining codes are called exons, and they will be spliced together. The new messenger RNA is now ready to exit the cell. Once in the cytoplasm of the cell, the messenger RNA will bind to a small ribosomal unit. This small ribosomal unit came from the nucleoli of the nucleus. Once messenger RNA binds to the small ribosome, the process of translation will begin. In the surrounding cytoplasm are large amounts of amino acids that become attached to another type of RNA called a transfer RNA. Transfer RNA looks somewhat like a hairpin. Attached to the base of each transfer RNA is a sequence of 3 nitrogen bases called anticodons. At the other end of the transfer RNA is an amino acid that corresponds to that anticodon. The anticodon is complementary to a specific codon on the messenger RNA and will deliver the amino acid to that specific codon. Thus, it is the codon on the messenger RNA that determines which amino acid will be used in that particular location. For example, the anticodon sequence AUG corresponds to the amino acid methionine. In cells of humans, all start codons will code for the amino acid methionine, so all start codons will be AUG. The start codon is the starting point on the messenger RNA for translation to occur. A tool called the messenger RNA wheel can be used to determine what messenger RNA will code for what amino acid. Using this wheel, if you start at the nitrogen base at the center of the wheel and work your way out, you can determine what amino acid that messenger RNA codon will correspond to. For example, the codon GCG codes for the amino acid alanine. The codon GCC also codes for alanine, as well as GCA and GCU. AAG, however, codes for the amino acid lysine. The transfer RNA molecule is a vehicle that brings the amino acids to the messenger RNA strand. The anticodon of the transfer RNA is permanently attached to it, but the amino acid is removable. When the amino acid is absent, the transfer RNA will float through the cytoplasm and attach itself to its matching amino acid. When transfer RNA delivers the amino acid to the messenger RNA strand, it can detach from the amino acid and return to the cytoplasm to search for another matching amino acid to bind to. At the start of transcription, the small ribosomal unit attaches itself to the start codon on messenger RNA. The small ribosomal unit has two binding sites. The transfer RNA with the complementary anticodon will bind at the start codon on the messenger RNA. The start codon again is always AUG for humans. The first transfer RNA is attached to the anticodon UAC, which is complementary to the start codon AUG on the messenger RNA. This first transfer RNA carries the amino acid methionine. Binding occurs between the complementary base pairs of the codon and the anticodon. Once the small ribosomal unit is attached to the messenger RNA, a larger ribosomal unit will attach to it using another type of RNA called ribosomal RNA. At the same time that the ribosomal units combine, a second transfer RNA arrives. This transfer RNA contains the anticodon that is complementary to the second codon on the messenger RNA strand. This transfer RNA carries the amino acid that corresponds to that specific anticodon. The anticodon binds to the codon. The two amino acids from these first two RNA form a peptide bond, and once the peptide bond is formed, the first transfer RNA is released from both the amino acid and the start codon on the messenger RNA. The ribosome now moves down one codon so that one of its binding sites still holds the transfer RNA in place, but there's now an empty binding site at the next codon. A third transfer RNA will bind to this next codon. Again, the anticodon from this third transfer RNA will be complementary to the codon on the messenger RNA, and once again the third transfer RNA will carry the amino acid that corresponds to that anticodon. Peptide bonds will be formed between the second and third amino acids, and the second transfer RNA will be released. The ribosome will move down one codon, and another transfer RNA will bring in another amino acid, which will bind to the growing amino acid chain. The chain of amino acids, called a polypeptide, will continue to elongate until the ribosome reaches the stop codon on the messenger RNA. The stop codon triggers the small ribosomal unit to detach from the large ribosomal unit and the messenger RNA. The completed polypeptide is released from all the components of the translation process and is now available to function in the cell as it was designed to do, either as a structural protein or as a secretion or as an enzyme. This ends Module 5 of Chapter 3, Transcription, Translation, and Protein Synthesis.